All right. I hope you all had a good lunch. And yeah, I just want to take a moment to introduce Guillaume here. I'm really excited to have him as an instructor. Guillaume luckily was able to come. He just arrived this morning. Was it from Barcelona?
No, from New York.
From New York, okay.
Barcelona the day before.
The day before, oh yeah, no big deal. Okay, so hopefully the jet lag, you'll be able to power through. And I have been working with Guillaume for years, we all have. The promise of NWB and Dandy has been that with standardized data formats, you can build standardized analysis tools and accelerate scientific research. And I think the Pineapple package is, in my mind, one of the packages that most helps us deliver on that promise, right? So it's a really wonderful symbiotic relationship between standardized data and standardized analysis pipelines. And it's, in my view, exactly what the field needs as a standardized framework to help us build good analysis pipelines quickly. So Guillaume's been part of this school for a while. I recently went to the Flatiron workshops where they teach these skills, and I was really impressed by the combination of lectures and interactive sessions. So I've asked Guillaume to come and do some of that here with us, and extended the time so that he can really show you not just the concepts, but how to write and run the code to actually do it. So I'm really excited to see your talk. Thanks for coming, Guillaume.
Thank you, Ben. So I'm very happy to be here. It was a pleasure to come here. I've seen many familiar faces of people that have come to our workshops, one here, but actually they probably left because I've already heard this talk. So all the faces I see here are kind of new. All right. So can you hear me okay? Yes? Okay. So I'll talk about Pineapple, and actually I will talk more than that. I will also talk about other packages we developed at Flatiron, kind of giving you an overview of what we do. I'll be here until Friday, so if you have any questions, if you want to try Pineapple on your dataset, please let me know. All right? So first question, where does Pineapple come from? Okay. So the package is quite new. It's like 5 years on GitHub now, but actually it's much older. So the ideas behind Pineapple are much older. And to explain that, I propose you this little graph here, which is basically, I've shown here on this slide, two types of experiments, okay? On the left, you have a type of experiment that I used to do a lot in my postdoc, which is basically sleep recordings. It was a sleep recording in rats, so the rats were implanted in the hippocampus and cortex. And as you can see, the experimentalists, they gather a lot of time series, so LFP, firing rate, spike times, okay? And on the other side, you have another type of experiment I have worked with on my PhD. It's more like a reinforcement learning type of task, where you basically have a stimulus, an action, a response, okay? So I propose that basically you can classify experiments in neuroscience in two categories, less structured and more structured. If you ever work with sleep data, you know you put the animal to sleep, the animal might sleep 1 hour, 2 hours, it might wake up, there will be noise, and so on, okay? So to deal with this kind of data set, which are basically very long time series with like timestamp time series that you have to synchronize and manipulate and remove the noise. The first author of this study, Francesco Battaglia, along with David Reddish, when they were postdoc, I believe, in Bruce McDonald lab, started a toolbox called the TS toolbox, so TS for timestamp toolbox. So the first version was in MATLAB. It was many decades ago. And Francesco Battaglia was a PhD supervisor of my postdoc advisor. So when I started in my postdoc lab, I was given the TS toolbox on a USB stick with no documentation, basically. That's how I started my postdoc. So I'm not very a MATLAB guy. I'm more like a Python guy. So I started very quickly rewrite a lot of functions on top of a new toolbox that Francesco had made, NeuroSeries. And so after a few years, a lot of people in the lab started to adopt the function I was writing. So we decided to actually make a new package that would basically encapsulate all of that. And so came Pineapple out of this, all right? So again, new package, but very old ideas on how to work with time series, timestamps, and epochs, okay? And that's what I'm going to explain to you here. Like at the heart of Pineapple is time series representation. Make sense? Yes? Okay. So next question usually is, when do I need Pineapple? Again, like this depends a lot of your data, but I will try to give you an idea. So another arrow, this time vertical. And here I'm going to show you what I would say are usually the steps in an experimental lab where you go from gathering some data. So here you have a mouse with a miniscope in a maze. And then usually what you do after you have basically gathered some data, you run some pre-processing pipeline. So for example, Kymon, Spike interface, or something else to kind of extract something out of the data, so the spike times, for example. And then you usually do some analysis, and then you can even do more very fancy analysis like a GLM, manifold, and so on. So Pineapple live in this little space here. There's a little space between the pre-processing and the post-processing where you take your data, your raw data, you transform them, you extract the relevant time series, and you want to align everything together. Basically, this camera gives this video, this prop gives this spike times. I want to put everything in a single format representation that I can basically manipulate, do some data wrangling, and move on to the next step, which is usually a model or some very fancy analysis. Okay, so that's the little space where Pineapple lives, basically. Does that make sense? Yes? Okay. So I will explain that also. All right. So let's dive into the package, because the idea is also that you might want to try it this week. At the heart of it is time series data. We do a lot of time series recording in system neuroscience. Okay. So for that, we have 3 objects to represent them. And I will start with the TSD for time series data. I hope it's obvious. Let's start with a simple example. Patch clamp recording. You patch a cell. You record the membrane potential over time. In Pineapple, this will appear like this in your terminal. You pass the time of the stamp, or the timestamps, the data. And what you get out of this is a nice representation similar to Panda series, if you are familiar to Panda, where you have the first column, which is the time, second column is the data. Okay? In Pineapple, we care a lot about a representation of the object. So there's nothing worse than trying a library and you can't make sense of the object you are manipulating, basically. So it always tries to give a nice representation. So under the hood is basically leveraged NumPy. If you have done any Python analysis in your life, you probably know NumPy. So we are built on top of NumPy, so nothing fancy, super stable. You can access the timestamp. You can access the data, okay? But also it's object-oriented approach, so you gain a lot of things. Automatic handling of time needs, epoch restriction, binning, time support. I will explain all of this, all right? So that's the first object we have. Second object, let's say you record your mouse in a 2D arena, you record the X and Y position, okay? So that's 2 time series that you can stack together because they have the same timestamp. So you can build it like this as a TSD frame where you pass timestamp, the data, 2 columns, and you label the column, and it gives you this table here which has the time and the data. Again, very similar to Panda data frame, if you are familiar with this, all right? Next example, TSD tensor. So we sometimes collect data more than 2 dimensions. Example, a movie. For example, here is castle imaging data. I have never found an example of something more than 3D, but if you have an example, let me know. And basically, you will use a TSD tensor. So anything more than 2D will be thrown in a TSD tensor, all right? So you have this kind of representation of time series, 3 objects to capture any time series. One of the differences with other libraries for neural data analysis is that in Pineapple, we don't specify what the object we receive. We don't label the object, LFP object, like a movie object, and so on. We have 3 objects like this that can capture any time series in a STEM neuroscience, okay? Which makes it more versatile. Make sense? All right. So next, doing math. So as I say, we are built on top of NumPy, so then it's very easy to extract NumPy value if you want to go in and out of Pineapple. But you can also apply any NumPy functions, which is very convenient. They have an approach for this within NumPy. So I will show you a few examples. Let's say you record a movie. So you have like 5 frames, 4 by 3 pixels. And let's say you want to have, for example, the average of the pixels across times. You can literally do np.min, the TSD tensor axis equals 0. You get out of Pineapple, you are back in NumPy. So now you have an array of 4 by 3 average pixel value. Now let's say, for example, I want to have the average value along the second dimension. So this dimension here, I can do np.min TSD tensor axis equal 1. And what do I get? A TSD frame. So Pineapple is smart enough to know that when you apply NumPy functions, depending on whether or not you keep the time axis or discard the time axis, you either go back to NumPy or you stay in Pineapple. Make sense? So it's like all of this is basically born in Pineapple, so you don't have to think about it. You can do slicing also. You can take the first frame here and you will get a NumPy object. You can get the first 2 frames, you get a Pineapple object, okay? So it's also smart about slicing. And you can also get, here you get a TSD transfer to TSD, so you stay in Pineapple, all right? So that's it for the time series with data. Now, in neuroscience, we collect a lot of data which are time series without data, aka a timestamp object. So you can think of, for example, the time at which the light turns on or the time at which the neuron spikes. So we have an object for this that we call the TS object. So the example I gave here is a mouse getting a reward and you want to know when the mouse is leaking. That's just a timestamp, basically. You instantiate it like that in Pineapple. NAP.ts give the time and you get a timestamp object. Does that make sense? Also interrupt me with questions if you have. It's going to be a kind of a long presentation, but please ask questions. So we have the TS object. Now, sometimes we record multiple timestamp objects. And so example here, of course, is a population of neurons activity. So you have 3 units here, for example. You want to group them together. Of course, what you need is a TS group of timestamp objects. So here you see a TS group, you pass data, you get a table. So I will explain that more into details because this is also one of the heart of Pineapple, like the core things of Pineapple. So collection of timestamp TS group object. It is built on top of a dictionary. Basically, you make a dictionary of timestamp objects because, of course, timestamps are like different times, so you can't basically put them in a TSD frame. Like that doesn't make sense. So you have to keep them isolated. So that's why we use dictionaries. That's how you create it in Pineapple. You make a TS group object like this. You pass a dictionary. Every row here is a TS object. And what you get is this table where every row here is a neuron. Okay. Now, the good thing that you can slice it, as you can show it here, but also you can apply any functions to the TS group object. And here I'm showing you one of the core functions of Pineapple that we use in every of our notebook. If I use in every of my analysis, I got my spike times. I want to bin the spike train. I want to count the number of spikes per time bin. Okay, so you can do tsgroup.count bin size equal 2, so 2 seconds, time unit equals second. And what you get, you go from a timestamp to a time series. What's a time series? It's a TSD frame object. So here you go from a Pineapple object to another Pineapple object, which has the center of the bin and the number of spikes in each bin. Okay, that's this example is basically random, but that's the idea. Okay? Yes?
Can the index just be
No, it can be only integers. Only integers, yeah.
This neuron elements is the long time series?
Yeah, so here, I think this one, no, actually, I forgot. It has actually 2 spikes per 2 seconds, basically. Yeah. These are just a random example that I made. But, yeah, so here, another question? It's fine. So, okay, so here is literally my little speech about code sharing. So here, we are here for data sharing. We share data, and we want to analyze it. But sometimes people don't talk about code sharing. Maybe it happened to you, you start in a new lab, you got shown some code from some postdoc that left years ago, and you have to figure out how he's indexing and slicing and how he's manipulating his data, basically. The advantage of Pineapple, and if you use Pineapple in your lab, is that basically you have a common way of representing data. So then when you arrive at this line in your code, tsgroup.count bin size equal 2, you don't have to figure out what the author of the code did. You can just read it as, oh yes, of course he's binning the spike train in 2 second bins, okay? So think about it if you want to adopt Pineapple. It's very important for code sharing, because code sharing always comes with the data. Then it can be very easy to read the code as if you read the method of a paper. I will show you more examples that hopefully convince you that basically the syntax of Pineapple is very easy to read and can be very, very, like, compress your code for doing analysis, all right? Any question? No? All right. So, so now we have 5 objects. We need one more in STEM neuroscience that we basically something we do a lot, which is basically measuring the start and end of times, aka epochs, all right? So here I show an example of stimuli grading, basically. Let's say there's the start and end of when the stimulus is presented. In Pineapple, it's called an interval set of intervals, and that's how you create it here. You give a list of start and list of end. So I'll talk about that again. Intervals of time, the interval set object. So I give a few examples. Sleep-wake, so classical one, when the animal starts sleeping, when his hand sleeping. Stimulus on-off, leak start end. And that's how you create it So the natural way of representing epochs is through a table where you have basically a list of start, a list of end. And in Pineapple, it shows like this, where you have basically, you pass the list of start, the list of end, and you have every row is an epoch. So in Pineapple, epochs are strictly non-overlapping. It makes things much easier, so they're strictly non-overlapping, okay? So now we have epochs, we have time series. Now I'm going to talk about one of the workhorse function of Pineapple, aka the restrict function. Okay, you get your data, you load your data. There are noise in your signal. There are some part of the data you don't care. There are only some subparts you care. You define interval sets, and you only want to analyze whatever is in those interval sets. So then what you can call is basically restrict. So I will show you that. So let's say, for example, you have those spike times from these neurons here as a TS object. Those are the epochs at which basically something is happening, and you only want the spike times within those two epochs. You can say TS.restrict ep, and you get only the spike times within those epochs. Okay. Again, think about code sharing, readability of the code. You arrive here. This is very, very easy to read. All right. Make sense? Okay. You can also do more complex manipulation in time. Basically, you can have interset object interacting. So I'll show you a few examples. So let's say you have two sets of epochs. So what you can do is basically the union. Let's say, for example, this is when the stimulus is on, and this is when the animal is awake. You can do everything together as a union of it. You can do the difference. Let's say, for example, this is when you have noise in your signals. You want to remove it. You can say epoch 1 set diff epoch 2. And for example, let's say, for example, this is when animal is running and stimulus is on. You can do the intersection of the two epochs like this. Okay. So again, manipulation of time interset can basically interact with each other. Does that make sense so far? Like I basically dive into the Pineapple object. That's kind of a summary slide, 6 objects. And the idea that with this, you capture basically any system neuroscience data set. Okay? Does that make sense? Yeah, all right. So we capture system neuroscience data set, but usually there is a lot of extra information that we have about our time series that we also will want to basically capture, and that can be helpful to do the analysis, all right? So I'll talk to you about metadata, data that comes on top of time series. Let's go back to this example of my TS group object. I have 3 neurons, one from thalamus, one from CA1, one from cerebellum. I kind of want to carry this information in my analysis script. Like I'm loading my data, and I want to know which neurons come from what probe, which group of channels, and so on. You can do this very easily in Pineapple by adding the field metadata. And here, basically, it's up to you what you specify. Here I say location, and I give some information about those neurons. And now I have this nice representation where each basically row is labeled of where it comes from. You can add anything usually in my script. I load my data. I compute some metrics about my neurons, like, for example, how they are tuned to something. I add this tuning measure to my metadata, and I filter after that. That's what I'm going to show you after. Let's say, for example, I computed some metrics. I give them some direction metrics, and basically it appears like this. I can also do it after creation of the object. In my script, I start doing some metrics, and then I create new metrics, and I want to add it to my TS group. I can do it after. All right? So you can do it for TS group. You can also do it for interval set. So here, for example, there's information about the orientation of the gratings. That's something I might want to carry on. So I can also specify metadata, and I have some trial tabs, for example, and I will appear like this. Okay? So you can extend this to infinity and have as many metadata as you want. And TSD frame, so those 3 objects, basically TSD frame, typically calcium transient, every colon is a neuron. Basically, you want to label, for example, which neurons, where did it come from in pixel space. So you want to carry that inside your TSD frame object. And it appears like this. So, okay, in this example, I give colors. Basically, I color every of my column. And I have this extra field here, give me some colors, all right? And so the cool things about it is that you can access it like this, but you can also slice it. So I'm going to show you an example here. Let's say, for example, I want all the neurons that are in the thalamus. I can do TS group.location equal thalamus. Again, code sharing, easy to read. Okay, I have a TS group object. I take only the neurons in the thalamus. Make sense? Okay. And I can make more complicated things. Let's say, for example, in this case, I want all the neurons which rate is above 5 Hertz and which direction is equal 0.3. Okay. So all of those lines usually appear at the beginning of my analysis pipeline. I take my data, I filter my neurons, I bin the spike train, and I do some complex analysis. Okay. Make sense? All right. So just a summary slide. And so that's it for the object. We have our object, we have some metadata. Now we want to do some analysis that are usually not the type of analysis we find in some classical library like NumPy, more advanced things. I will not go over everything because there's a lot of function in Pineapple. Just to give a few examples. So we saw the restrict functions where you pass basically an epoch and you get only the data within the epoch. Count, for example, we also saw it, count the timestamp. There is value from also, which is basically the basis of a tuning curve. And threshold also that give you anything above or below some threshold. That's just a few examples. There are more than that. So if you go to pineapple.org, you can actually find the whole list of core methods where basically you take those objects and you have them interact. And depending on the function you do, either you create a new object or you compute some metrics. All right? Make sense? Yeah, okay. Into summary slide. From any type of data in system neuroscience, basically I've shown you 4 examples here, to a few number of objects that capture all of them, to some methods to make them interact. Okay, that's how we are now. Now there is a question: is how to go from here to here, and that's why I want to talk about Pineapple I/O. So this is a very difficult problem, and I'm very happy I'm not the one addressing it, but those guys here are doing it. So I want to talk about, of course, of NWB. In system neuroscience, we are very unlucky. There's a lot of data format coming out of many different systems. It's a kind of a mess. So when we made Pineapple a few years ago, it was very clear that we did not want to basically cover all the possible formats to load in Pineapple. And thankfully, the NWB team has solved this issue. They have created a standard for sharing data, the NWB standard. So I guess you already had talks about NWB, so I'll just go a bit fast here. I just want to show you how to load NWB with Pineapple. That's the part that we really care about. You are in your lab, you create an NWB file, or you receive an NWB file. What can you do with it? And in fact, it's very easy. You pip install Pineapple, you import Pineapple as NAP, and you just do this. So this is my NWB file on my disk. I say NWB equal nap.load file my NWB. And what do I get? I get a table, basically, like this, which shows me inside the NWB file every object compatible with Pineapple. So NWB is a hierarchical data format. Okay, it contains a lot of information about the experiments, the experimentalist, and so on and so on. But Pineapple is a time series representation library. So what it does is that it flattens everything, looks at every object. Is it compatible with Pineapple or not? If it's compatible with Pineapple, it gives you an entry in the table here. Okay. So that's a very easy way to go from NWB to Pineapple and start your analysis. And so then what you can do is this NWB, for example, position time support, it gives me an interval set here. So, yes?
How does the hierarchy show up here?
It doesn't show. It flattens the hierarchy. It flattens the hierarchy. Yeah, so it's like, it just goes through. So there is a cool function in NWB that flattens it, goes through every object. Some objects are basically metadata information. You throw it away, you just get a time series or the interval set. Because here you want to do an analysis, so all this information is stored in NWB. Here it's all about basically things that are compatible with Pineapple. That's the idea. Okay? Makes sense? So literally, if you have an NWB file on your computer right now, you can just do pip install Pineapple, do this, and you can get started. All right? I don't think it's possible, right? NWB cannot have 2 keys. They can be no, it can. But yes. So actually, we saw this issue last year where we actually append something to the name here to disambiguate them. Yes, I remember this issue was solved already. Yeah. All right. Any question? No. Okay. So I keep going. So okay, now we are here. Data. I strongly suggest to make NWB because we are sharing data now, and it also makes it easier after that to load into Pineapple. All right, so that's a slide now where we are. Next step, standard analysis in STEM neuroscience. So here, the idea of Pineapple is not to provide you with all the cutting-edge analysis that you can see in the most recent papers. It's actually quite the opposite. It's all about the standard analysis that people have been doing in the field for like decades that we want to standardize in one library. That's the idea, okay? That's what I'm going to show here today. So to show you that, I'm going to give you one example of an experiment. So we simulate an experiment. The experiment is a mouse running on a circular track. There is a probe and there is a reward. So it's kind of a classical experiment in system neuroscience. And the data will appear like this, basically. You have some LFP, you have some spiking activity of 3 neurons, you have the position of the mouse on the circular track, and you have some reward time, and you have some 2 epochs here. I would say it's kind of a classical standard data set in STEM neuroscience, or at least is the one I'm going to demonstrate what we can do with it. So when you have this, first of all, you can represent that in Pineapple in a few number of objects. So you have the epochs here, the LFP as a TSD object, the spike as a TS group, the feature as a TSD, and the reward here as a timestamp. Okay? We start from here. What can we do? First question, the first question I will ask is what is the firing rate of my neurons as a function of my feature? That's okay, a tuning curve. Okay? So in Pineapple, it's a core of one function. You can do nip.compute tuning curve. You pass the TS group, the feature, number of bean, epochs, some range, some feature names, extra parameters, and give you basically the firing rate of each neurons as a function of the feature. 1 dimension, 2 dimensions, 3 dimension, any dimension, basically. Okay? First question we can ask. That makes sense? Yes? Okay. Second question we can ask, I forgot to say, in a recent year, we switched to XArray, so now the output is going to be an XArray object. It's actually going to look like this. If you're not familiar with XArray, it's a library that is very good at labeling dimension, basically. So it will look like this, where you have the firing rate for each neurons on some extra features. Second question I would want to ask is, what is the firing of one neuron relative to the other neurons, aka a cross-choreogram? So you can do nip.compute cross-choreogram, you pass the TS group, bin size, window size, epoch, and what I get is a Panda data frame where each column is a cross-choreogram between 2 neurons, and I show it for like a pair of neurons, neuron 0 versus neuron 1, which tells you that whenever neuron 0 fire here, there is a lot of spikes of neuron 1 after. So this relationship here is captured, okay? Is everyone familiar with cross-choreogram? Yes? Okay. You can do a peri-event, so I want to realign to a particular time. So here I realign to the time of the reward by a call of a single function, very standard, and I get a TS group object. I'm just going to sweep through this. You can do some decoding also. If I compute the tuning curve and I have some new epochs, can I predict the feature that I am observing? So you can do it in the call of one function in Pineapple, nip.decode biased for Bayesian decoding. You pass the tuning curve, the data, and you get a probability and best decoded position. You can do it in Bayesian decoding or template matching decoding, which are two approaches a bit different. Bayesian decoding assumes Poisson distribution. Template matching is neutral to the type of data you have. So if you have Casio imaging or spiking activity, it will work the same. All right? You can do a spectrogram. So this is a wavelet transform. So basically, if you pass your LFP as a TSD, you specify a bunch of frequencies, and you give you like a TSD frame, which gives you the spectrogram of your signal. All right. You can do a bandpass filtering like this. Apply bandpass filtering, some bandpass from some cutoff, and it gives you the filter signal. And so that's it. From this data set, with these few objects, you can already derive all of those analyses. From your experiment. Make sense? Okay. So the idea is that basically Pineapple has a hard wall on the right side of like what analyses are supported. The rule is that if your analysis requires to fit a model to the gradient descent, it should not be in Pineapple, because gradient descent are always annoying and a lot of tricky things, so it's not there. So anything that does not require gradient descent can be in Pineapple. So for the modeling, I will show you another package after. All right. There are more than that, but I will not show it here. There are more analyses. You can do other stuff. You can go to documentation online to see them. So that's kind of the overview of Pineapple, from NWB to a few number of objects to some methods to some standard analysis. You can basically write some code. It's a library to writing code in STEM neuroscience. Okay? All right. I will do some demonstration about visualization. Now we have the representation of the time series. And we have objects that are basically our time series, which are like a standard way of like aligning time. So one of the cool things that we get for almost free, that we can basically synchronize them, which is what I'm going to show you now. During my postdoc, it's one of the tools I wish I had. I was doing some recording, some cool sessions, and you want to basically browse the LFP, browse the spiking activity with the video, the camera that you have recorded, and you want to basically look at it all together. Also data stream, you want to look at them together. And so if you represent them in Pineapple object, The idea was to basically visualize them together. So that's the idea of PyNaVis. So it's a new package, Python neural analysis visualization with a few examples here. I think the best way for me to show you that is to actually demonstrate live. So I think I have time for that, right? Yes, I have time for this. So if you want to quickly try yourself, you can do pip install PyNaVis and just run this code, like do scope, and then they will work. But I think it's more cool if I show you actually on the terminal. So I will show you this package by moving my I can't do that. Hold on. Yep. No. Okay. Nope. I can't move this one here. That's annoying. Right. Any question about what I was talking about?
How are the epoch standards in the analysis? Like you can create, for example, cross parallelogram on one epoch and then you have
No, that's not possible. Like epochs, I mean, he will know about the epochs, but then
like you make the whole list with all the epochs combined and then
yeah. So you have to create your interval set with all your good epochs. For example, if you want to remove the noise, you pass it to your function, and it will basically jump every epochs. Okay, let me go back again. All right, here we go again. So basically, if you can do, let me just see, yes. Pip install PyNaVis Qt, and we get started, okay? It relies on something called PyGraphics, which is a library for like a visualization on GPU, which is beyond my knowledge, but it's very fast. But what I can do is this, PyNaVis, type PyNaVis and get started. And it's a GUI, basically. So then what I can do is this. I can go to File, Open, and I'm going to find an NWB file. So I'm going to go to this one, Data, and I'm going to pick Achilles, up, Open. And so what it's going to do is going to open the NWB and do what I just showed you, but like in the GUI, basically. So now if you have made an NWB file, or if you have an NWB file on your disk, you can start exploring, visualizing your data, basically. And so I have also objects. Can you guys see? Is it clear? Yeah. I have also objects that are basically all the Pineapple object. And if I double-click on one, it's automatically going to create the visualization for it. So here's basically my spikes from my NWB file created. And for example, let's say I have some EEG here, up, and I have the EEG. So it puts at the start of the recordings. And so basically I can play it up, and I can browse it, and so on. If I have, for example, I have some forward epoch. I should say those data are from the Buzaki lab. Basically, it was the data from, I forgot his name, but it was like epochal recording on a linear track. So forward epoch are when the rats run forward, okay? So we're gonna basically look at those forward epoch. And for that, I'm gonna go here, and I'm gonna say select interval set, and I'm gonna select the forward epoch here. And basically, it's gonna overlay the epoch on the signal, and I can just do that, and I jump to the first epoch, okay? So now, this is the time at which the animal is actually running along the track, and if you zoom back, you can start to see nice theta rhythm here, okay? There are many other things you can do. You can do a color by, group by, sort by, so it's all about you putting the right metadata. It acts on the metadata, basically. And so I can sort by the rate, for example. I can say sort by rate, up, and up, you will sort by rate, and I can say color by rate also, up, this one, up. Okay, so you can create cool visualization. You can jump between epochs. So one of the things I'm currently doing, for example, is that I'm doing a lot of dotted spike detection. So I have this LFP, and so I'm running my detector. I'm opening Pynavis, loading the epoch of the dotted spike to detect the LFP, and jump, and do some data quality control, basically. Is my detector the right one? All right. So that's one example of what you can do. You can do visualization like this. You can also open any movie. So I can go here. I can say file. Oh, merde. Sorry. No French cursing. I lost it. I'm sorry. Yeah. So I can go file. File open. I'm gonna find a video. Sorry, yeah, it's this one here. Let's say, for example, this is a video of your recording. So here it happens as an MP4 here, and I can do it here. And as you can see, this is my video of my mouse running in the environment. So now everything is synchronized, and I can play it, and I have the video, the LFP, the spikes, and all my epochs. So if you're also doing a lot of, for example, behavioral analysis, you run this pose estimation. You want to see if is the animal really doing what the pose estimation is telling you what it's doing. You can use Pynavis. If you are familiar with the package movement, for example, from the Sainsbury, that's one of the things we are trying to do this year is to basically integrate it so that if you have like a lot of ethological data set, you can actually watch it there. All right? Make sense? Any question? If you want to try it, let me know. I can explain you all the shortcuts. All right, and I'm going to stop for this, and I'm going to go back to the slide, and then I'm going to talk about modeling. All right? Yes?
Can I use remote? Do I have to download it?
Yeah, it doesn't work. Yeah. No, yeah. It has to be on disk, yeah. That would be cool, but that's also beyond my technical skills right now. Hop. All right. Okay, so we are here, okay. We are here, Pineapple. Know that, I hope it makes sense. Now, what about here? What's come after? So that's what I'm going to talk about, and I'm going to talk about this other package called NeMoS that we develop at Flatiron, all right? So modeling neural data. So for that, I will kind of give you an overview of what we do at Flatiron. We are in New York, near the Flatiron building, but we are not in the Flatiron building. We are next door. So this is the door of the Flatiron Institute. We are at the Center for Computational Neuroscience. And in this little axis, I've shown you where do we work and where we don't work. We don't work in data collection. We don't like Janelia. We don't collect data. We come after. Okay, so I just talked to you about Pineapple for data analysis and Pynavis. In the CCN, most of the people actually delve into this part of the axis, more like in the data modeling and model analysis. All right. So we have a package called NeMoS and a package called PlaneOptic. I will not talk about PlaneOptic because it's very, very beyond data modeling. I will talk about NeMoS. Okay. Make sense? Yeah. All right. So NeMoS for neural model, the address is here, Flatiron Institute /NeMoS. And it's a statistical modeling framework for STEM neuroscience. So, first question, which model should we do the first? Like, which one is the most classical in STEM neuroscience? And to us, the answer was a GLM, Generalized Linear Model, all right? Generalized Linear Model, also called LNP, where basically the idea is that you can model the response of a neuron as a function of a bunch of features. So you have your features here, you weight them, you pass through a nonlinearity, and you can predict a firing rate, okay? That's the idea of a GLM, Poisson GLM in NeMoS. For that, you have to design a feature matrix, which is basically time by columns. And so if I talk to you about time by columns, you immediately think about Pineapple. So, of course, NeMoS is compatible with Pineapple. The lead developer of NeMoS is Eduardo Balzani. Is my colleague, so we make the things compatible, all right? So that's how you do it in the code. You import NeMoS as NMO. You define the model like this. You define a GLM. By default, it's Poisson. You fit your data, so you pass your feature matrix, your spike count. And so you can predict the firing rate by passing the feature, and you can also score the model. If you have used scikit-learn, you can probably think, okay, but I can already use scikit-learn. I can do this in scikit-learn. Okay, there's nothing new here. So what is new here? So what is new here is what I'm going to show you after. Does that make sense so far for the GLM? who has ever fitted a GLM? Yeah? Okay, so you're familiar with it. All right. So what is very important in NeMoS is the basis, basically. We don't want to use the raw features to fit the GLM. We want to use a set of bases. So bases are used for feature construction. I show you a few examples here. Bases are this kind of curve here that basically you can put on top of your feature space. You have 1D, 2D, and log stretch for time. So bases are fixed nonlinearities. They are very, very nonlinear. And basically, they are used for avoiding over-parameterization, basically. So if I say, for example, you have like I was talking about this rat running on a linear track. Let's say it runs for 1 meter. I don't want to fit 100 weights for like each position in the track. I want to fit less weights that are gonna basically give you a smoother response, a smoother fit. So I can use basically basis to tilt the space of my linear track to learn only 10 maybe weight over the basis instead of 100. That helps the fit of the model. Okay, that's why you should use basis. But okay, having said that, why is NeMo easier for basis? That's what I'm gonna show now. So that's how it works in NeMoS. Let's say that's my linear track. That's my rats running around. Those are my bases stealing the feature space. Okay, in NeMoS, you can basically instantiate a basis function like this. You have your position here, and you can say basis compute features position, and you get 5 columns, which are basically the value of the basis at each time step of which the mouse or the rat run along the track. Okay, and so when you fit that, you get a better estimate of the position of the mouse. So you can get a 1D basis, but you can do more complicated things. And so, for example, let's say I have a 2D space. I have a rat running around, and instead of learning maybe 20 by 20 position, I want to learn maybe 5 by 5 only. I can basically have the basis interact, so I can multiply them here, and this is the code I can do. I can instantiate the first set of bases, a second set of bases, multiply them, and I get the interaction of the two. All right. So here the object X and Y are the time series position of the mouse. Those can be pineapple object. So NeMoS is smart also about pineapple. If you throw in a pineapple object, it knows how to handle it, and it gives you back a pineapple object. So it's very useful to compare time series when you plot everything. Okay. Now, even cooler, you can do the basis in time. So those are log stretch bases that were introduced by Pillow in his 2008 paper. Basically, the idea is that this is the most recent time, this is the most distant time, and the weight of the recent time should be more important than the weight of the distant time. So when you convolve with any feature that you have, you get basically recent time is more important, is more precise than the distant time. So that's how you do it in NeMoS. You create a raised cosine log conv, number of bases, and you can compute your feature. Okay? Makes sense? So if it's not very clear, the notebooks I shared in the Slack have a lot of examples of that where you can go through. So in the end, why NeMoS over, for example, scikit-learn is for this. I'm sorry, I should stop doing it. So why NeMoS over scikit-learn? Let's say you have 4 types of features, for example, some position, some velocity, some angular direction, and some spikes. You want to combine all of them together to learn a single model. You can instantiate a set of bases, add them together, and create this big feature design matrix. In NeMoS, it's a few lines of code, instantiate the basis, multiply them, another set of basis, basis plus basis plus basis, you get a new basis, and you pass all your features, and you're done. Okay? So that is not possible in scikit-learn, basically, or any other package. So that's why basically the advantage of NeMoS over other library is this few lines of code to create very complex basis, complex design matrix. Make sense? Yeah. Okay. So, few other reasons. It has Pineapple support, it's specific to neuroscience, it's compatible with scikit-learn, it's very fast, we rely on JAX, so we can run on GPU. There are a few observation models, Gamma, Gaussian, Poisson, Bernoulli. If you have calcium data, you can use Gamma or Gaussian. It has a bunch of regularization method, classifier. So what's coming next? So there's already a classifier GLM. GLM-HMM, so I believe now we have convinced Camila, I hope you all met Camila who is here, who was an intern last year, to actually give a presentation about the GLM-HMM. So maybe tomorrow or the day after, she will actually talk about it, and she can show you how you can use NeMo for GLM-HMM, and PGAM and point process GLM. So more models are coming. For now, we mostly have the GLM for neural data. All right? So kind of ending slides. Those are the packages we are developing at CCN. Those are the people working on the packages. Sarah is mostly working on Pineapple and Nemos. Eduardo, who is the lead developer of Nemos, and Billy, who is the lead developer of Planoptic. And those are the people basically that have contributed over the years. We are running workshops very regularly, basically. So we just did the last one at FENS in Barcelona last week here. So I believe the next one will be in Flatiron next year. And the idea is that you come for 2 days and we teach you Pineapple and Nemos with real example. We start with the data. We start with some tutorials. Then we take real data and we walk you through the notebooks where you can basically learn how to use those tools. And also, I would like to mention that we have internship every year. So you can also talk to Camilla. She was an intern last year. If you are interested in actually coming to Flatiron, either for software or research, there is an internship available. It's every summer. It's like from June to August, and you can work on any topics of your choice. All right. With that, that's my last slide. Thank you. Yes.
I just want to point out there's also a notebook that's
Yes. So you can also go to the Allen OpenScope, and there is a nice notebook that Camilla also made. But I think we're going to ask her to talk about one notebook only because she's going to help me. So yes, there is also another one on which dataset was it? I forgot. It was a GLM on the visual dataset, right? Yeah. On the Allen OpenScope, yeah. Okay, any other question? Okay, so Ben has asked me to give you some live demo. So if you're not too tired, I will take a 2-minute break, drink a little bit of water, and I'm going to give you like an actual example starting from real data, and we're going to show you what we can do. All right? Sounds good? All right, thank you very much. Okay, I shall restart. Okay, I will restart, and I think I have half an hour more, right? Ben, how much time? Yes?
Yeah, we have time.
Yeah, okay, all right. Yeah, so on the Slack, I share you this page, which is basically the answer to the workshop or notebooks we give. So basically, my idea here is that if, depending on what question you are doing, you can basically browse through that. I'll just go through the first one, maybe the second one if we have time. And otherwise, there are more. Basically, we try to cover many things. So we do I'm going to show you head direction cells, but there are like place cells. There is the Allen Institute visual coding dataset as well. So all of this is there. And so the notebooks have been filled already. All right. So I'm just going to run the cells basically and try to give you an intuition of how it works. Starting with here. So nothing interesting here. So here in this workshop, basically, we have packaged some data in some NWB file, MOS32 140822. The data are from Adrian Perrache paper and in the Buzaki lab, where basically he recorded head direction neurons in the anterior thalamus, so in mice. And so if you're not familiar with head direction neurons, there are neurons that fire for a particular direction of the head of the animal. So if I have one neuron firing from this direction, I have another one firing for this direction. And so those neurons fire for all possible directions, okay? So it's a very easy signal to track in the brain. That's why we are using it in this workshop, because it allows us to do very, very standard analysis, basically. So if I run this cell, the goal is just to give me my NWB file on my disk, okay? And so I'm going to use the function I was showing earlier, the nlp.load file. I have installed Pineapple. I give the path. And so if I do this, what do I get? I get a table. So this data has some spikes, has some slow SWS for slow wave sleep, which is basically the sleep, REM sleep, position time support, epochs, RY. RY is the rotation of the head of the animal in the horizontal plane, okay? So here is the idea that we're going to compute tuning curves. Make sense? That's our starting data. So next, what we want is actually to extract the data from the NWB, because actually this is not very loaded. It's just a preview of what's inside the NWB. Pineapple is smart to not load the data automatically, because sometimes NWB are very big, and so we don't want to basically load everything. But here I'm going to load it. So I'm going to call spikes. And basically what I get is this TS group object, where basically there is a bunch of neurons, and they have been labeled. So here you have the location, thalamus, ADN. Whatever metadata are in the NWB can be pulled inside the TS group object. So we try to capture as many metadata put in the NWB to be put in the NWB, in the Pineapple object, okay? So that's kind of an incentive to put as much as you can in the NWB object, basically. Group is the probe location of the neurons. So the recordings contain neurons from several brain areas, blah, blah, blah. So here, and I know already, I only want the neurons in ADN. So ADN stands for anterodorsal nucleus of the anterior thalamus. I only want neurons from that area, and I only care about the high-firing neurons. I don't want to deal with the low-firing neurons. So usually in all my script, I have this. I want to get rid of all the low-firing neurons. So I can do it in one line in Pineapple. Again, you can agree that this is very easy to read and understand what I'm doing here. So in one line of code, I'm going to filter my units, and I have actually less units now. So I'm just going to print spikes. And so what you see, I have less units with their rate, their location, and their group. So they come from three chunks, actually. Make sense? Okay, easy. So then I'm going to take the angle. So the angle is the head of the mouse. That's the data called RY. So I'm going to do it in one line here, and I extract the rotation of the head of the animal. We have a time series, which has the time stamps on the angle between 0 and 2 pi. Now here's the little subtleties of Pineapple. In very many cases, the data we are dealing with are very big, do not really fit into your RAM, so we don't want to load everything. If you actually print the underlying data, it's not a NumPy array, it's a HDF file dataset. It's actually a memory view of your data. So again, trying to be smart and not basically crash your computer. I want to load the epochs. So now I have 3 epochs. One was animal was sleeping, one was animal was running, and one was the animal was sleeping. So 3 epochs, one after the other, which is that tag here, sleep, wake, sleep. Okay. So again, all of this come from the NWB. All of this is pulled from the NWB metadata. Nothing is being created here. I'm gonna separate them in 2 categories: wake epoch and sleep epoch. So I'm gonna use their tags here, and I say epoch.tags equal wake, epoch.tag equal sleep. I run it, so I now create 2 type of interval set. And I'm going to call my first high-level function in Pineapple the compute tuning curve. Okay. So again, I want to know the firing rate of my neuron as a function of my feature. My feature is the rotation of the head of the animal. So I'm going to call compute tuning curve. I'm going to pass the spikes, the angle. I'm going to put 61 bins, the time support, which is basically when is a position defined. Run it, and boom, I have my tuning curve, basically, which are for every units and for every bin of the head direction, all right, in one line. So this works for any dimension. So if you have 1D, 2D, 3D, any dimension, I can compute a tuning curve. It can also work with TSD frames. So if, for example, I have calcium transient, you can also pass calcium transient here. It will work, okay? You can specify also the bins if we really want to be precise. Here, I just want to sample equally the feature space, all right? All of this is in the documentation, of course. If you read the docstring, you can read about that. Now let's plot it. Let's see if it's actually correct. Bunch of code. So here we show 2 neurons only. So neuron 7 and neuron 8. And those are basically the same thing shows in 2 different ways, in a normal way and in a polar plot. Those are the firing rate. This is the firing rate of the neuron as a function of the feature. And this is another neuron. And as you can see, when you plot it in polar plot, you get a peak in one direction. These neurons fire for this particular direction. These neurons fire most for this particular direction. They have kind of opposite direction, okay? Simple tuning curve in 1D. Make sense? All right. Now, just a bunch of code to get the preferred angle. So now I'm going to do what I said earlier. Halfway my analysis, I'm creating new metrics. So here I'm creating the metrics of what is a preferred angle. Dirty XRA code, we don't care. But what I can do now is basically add this information to my TS group, and by saying set info pref angle and get pref angle, and I create in these spikes in the TS group object a new column which basically tells me where is the preferred direction of these neurons. Okay, so again new matrix adding to the TS group carrying along your analysis. All right, make sense? So now in this TS group object I have all this column. And I'm going to basically now make a new plot in a very small interval set just to display it, just to convince you that basically those neurons encode for head direction. So now that I've made a raster plot where basically I have plotted in one single plot the head direction, and in another plot the firing rate of those neurons as a function of head direction. And as you can see, the firing rate of those neurons matches very well the head direction of the mouse. Okay. So it's a cool recordings, a lot of neurons we can do, and this can be done in a very few lines of code. I restrict the angle, and here spikes restrict the angle to TSD. I basically assign to every spikes the value of the preferred angle of that neurons, okay? Sorting them automatically. So I create a raster. Make sense? Okay. So now, next question, I want to know what is the relationships to each other. I want to compute cross-choreogram. So in one line of code, I can compute a cross-choreogram here by passing the spikes, a TS group object. I'm going to say in 200 millisecond bin size and for window size of 20 seconds, okay, only during wake. I run it. Well, I'm going to show it what it looks like. I run it. I have a Panda data frame where basically this is the time bins in the cross-choreogram, and this is every pair of neurons. So I don't want to display everything, it's a lot. So I'm just going to display 2 pairs that I pre-selected. And I'm going to try to walk you through this. So now basically I have 2 pairs, 7 and 20 here. One pair, 7 and 20, which is different colors. And as you can see, they fire for the same head direction, okay? The preferred direction. So if you compute that cross-choreogram, how many spikes of neuron 7 do I observe? How many spikes of neuron 20 do I observe whenever neuron 7 fires? You can see that they have a peak at time like 0. They have a lot of spikes in common. Now those 2 guys, 7 and 26, they fire for opposite direction. What happened in the cross-core? Whenever neuron 7 fire, there is no spikes of neuron 26. Makes sense? If ever this guy fire because my head is this direction, this guy won't fire. If my head is in this direction, this guy will fire, this guy won't fire, okay? So you can see their relationship, their functional relationship in the cross-choreogram, okay? Makes sense? All of this in 2 lines of code in Pineapple, and a few matplotlib functions. Now I'm going to do the same but during sleep. In this experiment, the animal was recording during wake and during sleep. So I'm going to call the same function but now passing sleep epoch. And now only the spike times during the sleep epoch are going to be computed. And I'm going to basically run the same plot. But now this time I have added a cross-choreogram during sleep. And what do we see? 2 neurons firing for the same direction have a peak in wake in the cross-choreogram, but also have a peak in sleep in the cross-choreogram. 2 neurons firing for opposite direction have a trough in the cross-choreogram during wake, but also have a trough in the cross-choreogram during sleep. Think about it. During sleep, the animal is sleeping, he is not moving his head, yet the activity of those neurons is maintained, the pairwise correlation is maintained as if it was awake, basically. This is basically the hallmark of the attractor dynamics. That was the reason why this paper was published and is a result from now actually a decade ago. And all of this in a few lines of Pineapple. Make sense? Does that make sense, this graph on this plot? Yeah? Okay. I will not show this one because this is basically more complicated stuff with NeMOs. Actually, yeah, let's just go through it. Yeah. So now that we have basically understood that those neurons have a functional relationship by looking at the cross-choreogram, we want to know if we can capture this functional relationship with NeMOS, basically. Can a model basically be able to predict the firing of one neuron based on the firing of other neurons, okay? So we're going to use the Poisson GLM for that. We're going to use NeMOS. So I'm going to run a bunch of code here just to basically preprocess the data. NeMOS always need to basically count the spike train and so on. So I'm going to do it a bit quickly. I'm going to define a sub-intervals of wake epoch to go fast. I'm going to define some bin size. So again, one of the central functions of Pineapple, counting the spike train. So here I define 10 millisecond bin size during wake, and I'm going to count the spikes during this wake epoch. I'm going to sort them by the preferred angle. And I'm going to basically restricting them again. Yeah. So here is just few functions just to explain what I'm doing. What we're going to do basically is convolving in time. So in NeMOS, you have those bases that convolve in time. The idea is that you want to try first to see what can you predict the activity of a neuron based on its past history. All right. So we're going to do this. This is the history. This is what you want to predict. So for this, in NeMo, okay, we have actually some cool animation. Is this going to work? No. Too bad. Yeah. Okay, it was just showing the convolution. Everyone familiar with convolution? Take a time series, you roll something over the time series. Yeah, familiar with convolution? Okay. So I'm just going to skip it. I'm just going to define basically a historic conv basis, which is basically taking a chunk of data in my past and rolling it like this over my data. Okay. I'm just going a bit fast here because it's not the most interesting. So if you were to come to a workshop, this is what you will have to work on, basically figuring out how to preprocess your data, put them in the right format, call NeMOS, and so on. So here I've created this design matrix, basically, where this is time and this is basically past bits of my histories as I move on in time, all right? We're going to fit a single neuron model. So just, again, some code given. So here's where basically things start to be interesting. I define a GLM basically from NeMOS, solver name equal ABFGS, and I'm going to call basically fitting the data using the input feature on the neuron count. All right. So I'm just up like this. Here, this is basically Pineapple object being passed to NeMOS. And NeMOS is smart to know that basically he should use them and fit over it. And so we have a GLM object being fitted, and basically we can plot the result, and what you have is basically a spike history weight. So now what we have learned is the weight over each basically bin of past activity. Okay. I can do it for the second half. So here I've split my data into two parts, and I have two fit, one fit from the first half of my data, one fit from the second half of my data. So this is cool, but it's a lot of basically weights to learn. Like I'm basically using a lot of points. Here I'm going to use a kernel, like a basis, that basically compress the past activity. So I'm going to use log stretch basis that looks like this. So I'm going to define them here. Raise cosine log conv. I define 8 bases that stretch sometimes in the past. And I'm going to basically say neuron count, compute features, the basis, and I'm going to convolve the spiking activity with my basis, basically. And so I'm going to define some epochs. And so this is what it does, basically. This is your spike times. So when you roll your basis over your spike times, it creates this time series where basically some bases are going to have a sharp response. Some bases are going to have like a delayed and big response, okay? So the more you increase the spikes, the more you're going to have some different weight over your past history depending on your basis shape, all right? I hope it makes sense. Now, I'm going to fit another GLM, basically this time using this reduced dimensionality problem, using some basis. And now I have a new GLM. I have some coefficients. All of this is given here. It's not very interesting. It's all about reshaping the coefficient. But what I have learned in this, basically, what I want to show you is this. Sorry, I'm just going fast here because I just want to explain to you what we can do, basically, in NeMOS. Yes. So what we have learned now is a new way of learning, is basically predicting the activity of the neuron based on its past history. This is not the cool figure I want to show. What I want to show is this one, all-to-all connectivity. I have a bunch of neurons, and I want to predict the activity of my neuron based on all the other guys, basically. That's the problem I want to do now. So I'm going to do the same by basically taking my basis, passing all my spike count of all my neurons, and creating a big design matrix, which is basically activity of all the neurons, and fit a population GLM. So in NeMOS, it's an important point here that I didn't mention in the slide. We have GLM and we have population GLM. Again, we tailor for system neuroscience. So if you have some neural data with a lot of neurons, instead of fitting one GLM and calling a multiple times the GLM, you can just pass your data in a population GLM, and it will do the same. So here I'm passing all my neural data of all my neurons, the convolve count, and I'm learning a population GLM, basically. And I have a new model, which doesn't fit very well, but it's okay. I can predict the firing rate. So this is what the predicted firing rate looks like. So here, basically, this is an animal head direction. This is a neural activity of all my neurons, okay, that tracks the head direction. And this is what I can predict from NeMOS. Here, think about it, I did not give the head direction, I just gave the activity of the other neurons to predict the activity of one guy. With that, I'm able to reconstruct the particular head direction of the population, okay, without any knowledge of the feature space. But that's not what I want to show. What I want to show is this. I'm just slicing the weight together. And this is what I want to show. So now what I've done is that I have 18 neurons. They're all tuned to head direction. And what I've learned is this big matrix, which shows me what is the weight of the GLM for predicting the activity of another neurons, okay, sorted by their preferred direction. So those neurons have been sorted by their preferred direction in the horizontal plane. And basically, this guy was predicted based on the activity of all the other guys. And so you get this nice gradient that shows you basically, if you are familiar with it, the Mexican hat connectivity. Guys firing for the same direction are going to have positive weight along the diagonal. Guys firing for opposite direction are going to have negative weight. So this functional connectivity matrix can be learned in one function, in one equal of one function in NeMOS. That makes sense what we are seeing here? All right. No. Yes, question. So population GLM is basically an extension of the GLM in the sense that when you call fit here, so when you call fit here, count, so if you use GLM, count has to be a one-dimensional array. If you use population GLM, count has to be a 2D array where basically the first dimension is time, the second dimensional is neuron, which means that basically it fits iteratively every column separately. So it's just a faster way. It would be equivalent to writing yourself the loop of instantiating a GLM for each neuron that you have. But then if you have the same design matrix, you just want to go faster sometimes and say, for this design matrix, I want to learn all those neurons. And that's what it does, basically. So sorry?
So each neuron is still independent.
Yes. Yeah. So each one is an independent fit. Yeah. Just a more packed, convenient way of fitting a bunch of neurons independently. Yeah. And also there is some optimization behind with JAX that Eduardo is a very expert at. So yeah. Okay, does this plot make sense? So just one example of analysis you can do with NeMOS here. All right. Do we still have? I have 10 minutes. I'm going to show you one more things, and then I'm going to stop. One of the important things, because here I show you a lot of spikes, so people usually get confused. They think that basically Pineapple is mostly for spike times. In fact, it's not. You can also use it for Casio imaging. So if your data are Casio imaging, I just want to show you quickly one notebook where now we're going to look at head direction neurons in Casio imaging. Okay, same system, neurons firing for a particular direction, this time in Casio imaging, and this is what it looks like basically. So we're going to just run a few things, starting from some import. Load some data. So here I have another NWB file. Again, same thing. I load the data. I load the file. again, I have this nice table showing me basically all the data on ROI response series is the object in NWB for Cassium data. So now I have a TSD frame object, okay? So I don't have a TS group, I have a TSD frame object. TSD frame represents the transient of all my neurons. So here, every colon is a neuron. This is the time of my recorded activity. Quickly, just to see what it looks like. Here, I'm plotting you two neurons that are calcium imaging neurons. Those are the response, the transient response, okay? And again, I want tuning curve. I want to know the mean fluorescence of my neuron as a function of my head direction. I can call compute tuning curve. I pass the transient, the angle, same function, and it gives me also an X-array, okay? So now the computation internally is a bit different. It's not like counting, it's not a rate, it's just an average of whatever you pass. So here, delta F over F, like whatever units it is, it's just an average of that for every bin of your TSD frame. But then if I plot it, I can visualize it similarly. And now I have two units, for example, which also have tunings for a particular direction. Again, same type of data, different formality, different type of data. One thing I didn't show earlier, decoding. So here I'm going to take a small interval, and I'm going to call decode template, okay? So I completed my tuning curve. I have some transient. I want to see if I can predict the head direction based on my tuning curve. Call of one function, NLP.decode template. I pass my tuning curve. I pass my transient. 100 millisecond bin size, so basically look into 100 millisecond bins, and I'm going to use a metric correlation. Template matching basically tells you, given this bin of activity, where does it match the most in my tuning curve? Okay, so it basically does it minimize, it finds a minimum distance from your bin to all your tuning curve bins. Okay, so when you do that, you get a decoded angle, and I can plot it. As you can see, it does a fairly good job. So first of this is basically distance between the each bin in time here on my tuning curve. And basically you can see here, you can recover the true head direction. So in this one-dimensional feature, easy problem, of course, if you have a different type of feature might be not as easy, but the good thing about head direction is that it's very easy to decode when you have a lot of data. Here you can see for each bin, each time steps, I'm very good at actually recovering the true head direction of the mouse. Okay? All of this is a call of one function. And I will stop here, but you can also model some data with GLM. You can just need to change basically you don't use Poisson, you can use Gaussian or Gamma. Okay, observation model, sorry. Does that make sense? Okay, like, in the end, it's like, if you're someone in WB, you can get started and already do a lot of analysis with these tools, basically. Okay. Any question? Okay. All right. Thank you very much.